Hi, everyone. Welcome to our third AP Bio Chat. Following the structure from last time, what I did was I decided to focus on a single question and expand out from that question as much as you would need to know for the AP exam. So we're going to be unpacking the topic of enzyme inhibition today. Before we get started, just a little introduction. If you have any questions for me, my name is Lina. You can email me here. I work for Cold Spring Harbor. And we're offering a series of online resources through our website. So you can check all of those out by visiting this website. You can also check out our social media pages. Obviously, you're here on YouTube. You can also check us out on Facebook, Twitter, or Instagram. So we've discussed this, but in case you haven't joined us for the previous two chats, the AP Bio exam this year will only consist of units 1 through 6, which I've listed out by topic right here. Unit 7 and 8, because of school closures due to coronavirus, will not be tested. So no natural selection and no ecology. And then last Friday, April 3rd, there were some updates announced through the College Board to what the test will look like. So they're offering the test 2 times. The first time is May 18th. This is pushed back a week from what it was originally scheduled to be. So you have an extra week to study. It'll be at 2 p.m. Eastern Standard Time, and everyone will take the exam at the same time. So unlike in the past where like you would take it at 8 a.m. if you were on the East Coast and 8 a.m. if you're on the West Coast, everyone is taking it at 2 p.m. Eastern Standard Time. So if you're not Eastern Standard Time, you should look up what time that would be for you. If you can't make the May 18th date, there is a second administration on June 3rd at 4 p.m. Eastern Standard Time. They announced on Friday that the 2 free response questions would be as follows. So there will be a 1 25-minute long question and 1 15-minute long question. They announced some more details, which I have on the next screen. So the first question is going to be like lab-based, and it's going to be worth over half of your score. The second question is going to be based on some sort of concept and breaking that concept down. So really demonstrating your understanding and expertise on any concept from units 1 through 6. It'll be about 35% of your overall weighted score. So let's focus on question 1 first. The question name is interpreting and evaluating experimental results. So they'll give you some sort of experimental scenario, right? Some sort of research study, maybe a graph from a lab, maybe a data table from a lab. And they'll ask you to do the following. So they'll ask you to describe and explain the biological concepts applied in the context. So for example, if the experiment is about plant transpiration, you should be able to explain what is happening. The second thing they'll ask you to do is to be able to explain the experiment. So what that really means is you should be able to understand what the independent variable was in the experiment, what the dependent variable was, what were the controls that were used, what the control group was versus the experimental groups, those sort of basics. They're going to be asking you to analyze data and then make and justify predictions. So I actually came up with a question that fits this scenario since it's worth so much of your score that follows the enzyme inhibition topic. So we'll take a look at that later in the session. The second question is a shorter question. It's worth less of your score, but still important. It's conceptual analysis. And this is going to be similar to what we did last week, where I sort of just asked you to describe and explain what was going on with DNA replication. Some of the objectives they might have for you are to describe biological concepts or processes, predict causes or effects of a change in a biological system. So what this might look like, for example, for enzyme inhibition is, what would happen to the enzymatic reaction rate if you introduced an inhibitor? And then finally, they're going to ask you to obviously support or justify any arguments you make with your prior knowledge. Okay, so this is an overview. Are there any questions so far about the two different types of questions that you will see on the AP exam this year? Okay, great. So the question that I pulled for this week that will organize our lesson today is, okay, so follow up from the question we did on Tuesday, what types of inhibitors should we know? So I think the first AP Bio Chat, I'm really sorry for this, was like a huge compilation of all the questions I got on Instagram, and I sort of just touched on everything at face value without really going into the topics into too much depth. Um, I think for review purposes, it is useful to go into a little bit of depth, so that's what we're going to do today. The enzymes that you absolutely must know for the AP exam are competitive inhibitors and non-competitive inhibitors. So I'm going to tell you guys a little bit more about those. This is coming from, so if you do need to do more review on enzymes generally, this is coming from Unit 3, Objective 3, which is environmental factors that affect enzymatic reactions. So as a quick refresher, the three that you need to know for the AP are pH, temperature, and enzyme inhibitors. So NB asks, will I be able to use notes? So for the AP, I do not know if they made any announcement about your ability to use notes. I would imagine not, but you can check out the College Board website to find out. Sorry, I don't know off the top of my head. In any case, the common thread, so going back here, the common thread between these three environmental factors that affect enzymatic reactions is that all of them are going to make the active site more or less accessible to substrate or totally inaccessible. So you probably have learned about optimum pH and optimum temperature. Enzyme inhibitors are obviously going to make the active site less accessible. And then a molecular consideration with regard to the active site is what types of bonds or interactions are affected in each of these three scenarios. Importantly, you need to know that with pH and temperature, you're going to be affecting the hydrogen bonds. And then we'll talk about what types of bonds are affected in more detail when we talk about the enzyme inhibitors. So competitive and non-competitive are going to look a little bit different. But for each of these, you should always consider what types of bonds are involved when we're in the land of bio. All right. Any questions so far? Great. So like I advised you guys last time, I came up with a little list of words that I feel like are key terms. So these are things that I'm going to want to use as vocabulary or as evidence that I know what I'm talking about when I'm writing my free response questions. So the key terms for today are denaturation. Denaturation is when your enzyme unfolds due to changes in pH or temperature. Optimum temperature and pH, you should be familiar not only with the definitions here, so these are the temperature and pH at which your enzyme works the best, but you should be familiar with those graphs that look something like this. So you might have your axis here, and let's say that this is 37 degrees Fahrenheit. I'm sorry, Celsius. You might have an enzyme activity curve that looks like this. And what we can interpret is that whatever enzyme this is works best at this 37 degrees Celsius. You should know the terms competitive and non-competitive inhibitor. You should be familiar with what an allosteric site is, and so that's going to be connected to this guy. And then you should also be familiar with what a conformational change is. Okay. Any questions so far? Okay. So we're going to start with competitive inhibitors. I just created a really simple cartoon for us to talk about the topic. So the way I remember it, and I know your teachers are always like, don't use the word in the definition. But I think with bio, there's an exception because a lot of times these fancy names are exactly what they sound like. So competitive contains the word compete, right? So competitive inhibitors compete with substrate for the active site. So I'm going to just really quickly show you guys what I mean by that. So we have a pretty typical enzyme substrate mixture here, right? And in a normal reaction, you would have the substrate bind to the enzyme's active site. Notice how they fit together really well. And then the substrate undergoes the reaction and we have the formation of products, right? Successful enzymatic reaction took place. When you introduce competitive inhibitors, you'll notice that the way that I've drawn out this competitive inhibitor looks like it fits the active site like perfectly, right? So with the competitive inhibitor introduced, you now have this molecule that is competing for the active site. And if it wins the competition, if it actually binds to the active site, these poor substrate molecules can no longer be worked on by the enzyme. And if they can't be worked on by the enzyme, then your reaction isn't going to take place, okay? So competitive inhibitors compete with substrate for the active site. Therefore, the substrate can't bind the active site. And if the substrate can't bind the active site, your product cannot be made, okay? And so what happens here is you end up with either a slowed reaction rate or a completely halted reaction rate. Fun fact, a lot of like poisons and toxins behave as competitive inhibitors, halting enzymatic reactions. And so if poisons and toxins in our environment, right, are capable of this competitive inhibition, you probably are rightfully thinking that we probably want a way to rescue this competitive inhibition effect, right? So is it possible to rescue competitive inhibitor effects? So just think about this for a second. Is it possible to stop this competitive inhibitor from binding to the active site? And the answer to that question is absolutely, right? So how do we, and let's just think outside the biological world, right? If you have someone who's competing with you, how do you beat them? You become a better competitor yourself, right? So the strategy here from a medicinal standpoint, right, is if we are dealing with some sort of competitive inhibitor that is acting as a toxin or acting as a poison, what we can do is we can increase the concentration of our substrate. In other words, we get our substrate to outcompete the competitor, right? So I have a little diagram of how this works. It does look like a Pac-Man cat. Okay, so this is the mechanism for increasing substrate concentration to outcompete competitive inhibitors. So this is our like little scenario. Let's say that this like toxic competitive inhibitor has invaded our enzyme reaction system. If we give the patient more substrate, right, increase the concentration of substrate, all of a sudden, just from a probabilistic standpoint, from a statistical standpoint, if you have way more substrate than you do competitive inhibitor, the chance that a competitive inhibitor binds this active site goes down significantly. And so we can rescue competitive inhibition by increasing substrate concentration. That's a really important concept. And as we go through non-competitive inhibition, I want you to contrast the two sort of scenarios. Okay. Any questions about competitive inhibitors so far? Okay. So because substrate was able to bind, we have a successful enzymatic reaction. products are able to be made, and we're all good, right? Okay. So let's shift gears and now talk about non-competitive inhibitors. Non-competitive inhibitors are exactly what they sound like. So if you're not competing, right? If you're not competing, then you're doing something else, right? So non-competitive inhibitors are going to bind to what is known as an allosteric site. An allosteric site is a fancy way of saying somewhere other than the active site. So somewhere else. So we are binding somewhere else. Okay. So what happens when you bind somewhere else? So when you bind somewhere else on the enzyme, remember enzymes are simply folded proteins, right? If you have another molecule binding to your protein in another region, it can change the folding. A fancier way of saying this and of making sure you secure that 5 is by saying that we change the conformational shape of the enzyme, right? So a conformational change is a fancy way of saying a shape change. So if this non-competitive inhibitor, which I've indicated in red here, binds to the enzyme at a location other than the active site, what happened to the active site? What happened to the active site? It got a lot smaller, right? So look what happened to our active site. Our active site folded in on itself. It underwent a conformational change, and now the substrate is blocked. The substrate is blocked. It can no longer access the active site. And if the substrate cannot bind, are you going to be making any product? No, right? So there's no product being made. And so non-competitive inhibitors are going to present their own challenges because will increasing substrate matter here? So let's draw it out. If we increase the amount of substrate, is any of those substrates going to be able to bind to the enzyme? No, right? Because we've changed the structure of the enzyme. And as you guys have hopefully picked up on throughout your biology studies, that when you change the structure of something, you're necessarily going to change the function of it, right? And in this particular case, changing the folding of the active site makes this enzyme no longer functional, right? And so this can have some pretty catastrophic consequences on the cell, depending on how important this reaction is for survival, right? K. Evan Thompson says, my coworkers are non-competitive inhibitors because I'm the boss. Okay, I don't really get that, but okay. All right, and so I wanted to, as usual, wrap up the session with a sample free response question. So if you're joining us live, I'd invite you to read through the question and start thinking about it before we answer it together. If you are streaming this not live, what I would recommend you do at this point in the video is pause it and attempt to answer the question on your own on a separate sheet of notebook paper. So I'm going to give us just a moment here to read through the question before we start answering it. Okay. And I'm going to annotate the question the way that I would if I were taking the test. So it says HIV, the virus that causes AIDS, utilizes an enzyme called HIV protease to cut its viral proteins to the appropriate size. Those protein fragments are used to assemble new HIV viruses. All right. So before I continue, I just want to draw out a quick schematic of what this problem is telling us. They are telling us that this enzyme helps make protein. So it helps us, well, not helps us, it actually helps the HIV viruses It's helping make viral protein. So I'm going to call that protein subscript V. And that viral protein goes into making new HIV viruses. All right. At this point in your biological studies, you probably have seen much more complicated pathways than this, but I find it really helpful to diagram if they're giving you like a lot of vocabulary, a lot of terms. This one's pretty simple, but I find it useful to diagram out like a little pathway for myself. The reason I like to do that is because I can very easily see, right? If I knock out the enzyme, right? So if I get rid of the enzyme, I'm no longer going to be making protein. If I get rid of my protein, I'm no longer going to be able to make new HIV molecules, right? Okay, back to the question. It says a researcher studies HIV replication and a potential therapeutic, an HIV protease competitive inhibitor, and summarizes the data in a chart. All right, our chart is over here and we see data for, so minus, in case you haven't seen this in your labs yet, minus means you don't add it. So this means there's no competitive inhibitor. Over here, it says plus competitive inhibitor. So that means that there is, so that yes, there is competitive inhibitor in these reactions, right? Okay. And so if we look over here, we can sort of see that the HIV viruses without competitive inhibitor are increasing over time, which is reported in days. And then the HIV virus number with competitive inhibitor remains constant. In other words, we're not gaining any HIV viruses, right? And then, you know, this is conducted at 37 degrees Celsius. If at this point you are not familiar with 37 degrees Celsius, it's very important biologically. It's approximately the human body temperature. So generally we see that a lot of like bacteria and viruses have evolved to do things really well at 37 degrees Celsius. In any case, the trend is that, you know, we're increasing, moving down here, and then from here to here, there's no change. So we stay the same the whole way. And I guess this, we can make a little arrow here saying it's increasing. Okay. And then we can sort of start to look at the questions. The first is going to ask us to graph the data. That's pretty expected, right? Anytime you see just a data chart and no graph, you can bet that they are going to ask you to draw a graph for them. B says calculate the rate of HIV replication with and without the addition of competitive inhibitor. So they're going to ask us to figure out how fast this is happening. C says predict expected results if the scientists repeated the experiment at 40 degrees Celsius. D says describe how HIV virus replication is affected by the addition of competitive inhibitor. So this is going to be the part where they expect us to use our biology knowledge to tell us what's happening on a molecular level. And then E is going to be the question where they ask you to make a prediction and also like extend on D. So E says some of the most effective drugs against HIV are competitive inhibitors of an HIV enzyme. But many HIV strains have now evolved resistance to these drugs. Predict how these strains have evolved. Okay, that's a lot. But my general strategies for you guys, you know, for these complex enzyme biological pathway questions is to draw yourself a little pathway, start to analyze the chart, and then let's go ahead and start attacking these questions. So with the graph, you know, it's pretty straightforward. All you have to do is figure out what your X and Y axis is. Anytime you see a time course, time is always going to go on the X axis, right? And then in terms of the y-axis is simply going to be the number of HIV viruses. So this is going to be your y-axis, okay? All right, and then since we have two sort of data sets here, I'm going to do a line graph. Generally speaking, anytime you have change over time, it's best to do a line graph because the slope of the line tells you the rate of change, right? Okay, so we're using a line graph. And then here we're just going to do a multiple line graph because we've got two data sets, right? All right. This graph is a very bare bones representation of what you should do. I sort of just put something together, but you want to make sure you have your X and Y axis labeled. Something that I absolutely needed to do here, but was just frankly too lazy to do on my computer, is that you must include appropriate scales on your X and Y axes. So this should go theoretically all the way up to, I would make it to 30,000. So I always recommend going a little bit above the maximum. So since the maximum here is 25,000, I'd go up to 30,000 on my scale. So this would be 15,000, this would be 0, and so on and so forth. And then over here, I would go ahead and make this, up to your 5-day mark. All right. And then notice I have a little key for myself. So I have a line for with the addition of competitive inhibitor. So you sort of see that there's this flat line down here. And then without the addition of competitive inhibitor, virus is, it seems to be growing like exponentially, right? Okay, cool. For part B, they're asking you to calculate the rate of replication. So the way that you should do this, right, is by sort of asking yourself, what is changing from time 0 to time 1 to time 2 to time 3 to time 4? And the pattern that I noticed is that one, it's not linear, right? It's not a linear rate of change because it's not the same increment of change every time, right? We went up 300 on day 1, then the next day we went up 1200, right? So it's not linear. And in fact, graphing it helped us realize it's not linear, it's exponential, right? And so we need to figure out just how exponential it is. As it turns out, each day you are increasing quadruply, right? So going from 100 to 400 is an increase of a factor by 4. 400 to 1600 again is 4 times the amount you started with, and then so on and so forth, right? 1600 to 6400, you're increasing by 4. And then 6400 to 25,000 is an increase of a multiple of 4. So this is exponential growth. You are doubling, effectively, you're doubling twice a day, right? And they might ask it to you that way. They might ask you what the doubling time is. And in this particular case, if they asked it to you that way, if they said, what's the doubling time of the HIV virus? You could simply say, it's half a day approximately, right? Okay, and then in terms of the rate of replication here, you can just say it's 0 because no replication is happening. You have 100 the whole way through, so there's no replication. So the inhibitor is working, right? Okay, cool. Part C asks us to predict expected results if the scientist repeated the experiment at 40 degrees Celsius. So this is just a question going back to 3.3 of those environmental factors. If you increase the temperature, all right, like a couple things could be correct here. So what I would just say is that you could denature the enzyme and stop viral replication altogether. Another option that could be fair here is perhaps this is a type of virus that is thermophilic. Maybe it's evolved to work best at 40 degrees Celsius. We don't have enough information to say one way or the other. And so it could theoretically be okay for you to say here that 40 degrees Celsius would be the optimum temperature and viral replication would like skyrocket, right? Okay. But for my answer, I said denaturation. All right, D says describe how HIV virus replication is affected by the addition of a competitive inhibitor. Here it's saying describe. So it's not enough to just say adding a competitive inhibitor stops replication. You have to explain how that's happening. And so this is where the vocab is going to be important, right? You want to make sure you can use the word active site. You want to make sure that you can use the word, like stopping the reaction from happening, right? So what I said is, since viral replication involves an enzyme to occur, adding a competitive inhibitor stops that reaction from happening. This is because the competitive inhibitors bind to the active site of the viral enzyme. This prevents viral proteins from being manufactured. And then if you don't have viral proteins, then you can't make the viral capsid. This is like a little bit, I think, the difference between a 4 and a 5 here, right? You should know that a viral coating is made up of protein, right? And so if you don't have that protein coat, you're not going to be able to make any more viruses, right? All right. In any case, this is an answer that I think would be sufficient for getting a 5. And then for the last part of this question, it says some of the most effective drugs against HIV are competitive inhibitors of an HIV enzyme, but many HIV strains have now evolved resistance. Predict how these strains have evolved. Well, this goes back to the question of structure and function, right? So if the competitive inhibitors are binding to the active site and all of a sudden they don't bind anymore, then what probably has changed? The structure of the active site, right? And so the answer here sort of fits that narrative. So I went from all the way, from the amino acid sequence. So I said mutations altered the amino acid sequence located in the active site. The changes in the amino acid sequence must have resulted in the change of the conformational shape of the active site. If you don't remember like the conformational shape or conformational change, you can just say it changes the way that the protein folds. It's the same thing. So just you could say like protein folding, and that would be A-okay. Although being able to use these like fancier terms is a way to cue to the readers like, I got this, give me a 5, right? So you're changing the conformational shape of the active site. Since the conformational shape is different, a new competitive inhibitor must be designed to fit that new active site in order for the drug to be effective. All right, so this is my like 5 response. I'm gonna give the chat an opportunity to ask questions because I see that there's a delay in the presentation, but that's all that I have for you guys today. If there are any questions, feel free to leave them in the chat. If the chat closes for some reason, feel free to leave them as a comment in the question. Also, feel free to subscribe to the DNA Learning Center. We are hosting live streaming events to support students who are shifting to a learn from home model. I know that that's challenging, and we're here to support you in any way we can. So feel free to reach out in the comments, or you can reach out to me via email. Thank you so much for your time today. I appreciate it, and take care.